So, good evening, everyone. A very warm welcome to all the distinguished guests, respected teachers, researchers, students, and friends, and all nature enthusiasts who have joined us from the different parts of India. On behalf of Maharashtra Vriksha Samvardhini Pune, Department of Botany, Ram Niranjan Jhunjhunwala College, Ghatkopar, Mumbai, the Institute of Natural History, Education and Research, Pune, and Dr. Balasaheb Khardekar College, Bengurla. It is my great pleasure to welcome to you all to yet another exciting session on our monthly lecture series 2026. Our lecture series aims to bring together experts working in diverse areas of plant sciences and biodiversity research, providing a platform to share knowledge, inspire young researchers, and strengthen scientific collaborations. We sincerely thank each one of you for taking time out from your busy schedule to be with us this evening. Today's lecture is titled Cytotaxonomy, an underexplored discipline in the Western Ghats of India. And today we have with us Dr. Manoj Lekhak. It is indeed my privilege to introduce today's distinguished speaker, Dr. Manoj Lekhak, who is Associate Professor in the Department of Botany, Shivaji University, Kolhapur. Dr. Lekhak completed his BSc honors in botany from the University of Delhi, followed by his MSc in botany from the University of Delhi, and earned his PhD in botany from Shivaji University, Kolhapur, under the able guidance of Dr. S.R. Yadav, who is a well-known personality in the field of angiosperm taxonomy. His research focuses on plant systematics, cytotaxonomy, cytogenetics in the taxonomy of geophytic monocots and endemic flora of the Western Ghats. He has published extensively in leading international journals with recent contributions in systematics and biodiversity, plant molecular biology reporter, Journal of Plant Research, microscopy and microanalysis, and the nucleus, to name the few distinguished journals. He currently supervises 6 PhD scholars and has presented his research at prestigious international conferences, including the International Botanical Congress and the Asia Pacific Chromosome Colloquium. In recognition of his outstanding contributions, Dr. Lekhak has received several prestigious honors, including the Y.S. Murthy Medal Award from the Indian Botanical Society, and was also recently nominated as a member of the National Academy of Sciences, India. Sir, it's really honor to have you here on today's wonderful evening, and I welcome you on behalf of all the institutes who are collaborating for this uh, monthly lecture series. So I welcome you once again, and please start sharing your valuable knowledge to us regarding the cytotaxonomy. Thank you, sir.
Sir, is the screen visible?
Yes, sir. Make it full screen.
Is it visible now?
Yes. Yes, sir. Yes, sir.
So at the very outset, I would like to thank the organizers for giving me a chance to present my work, whatever we are doing in the Shivaji University, Kolhapur. I would also like to thank Professor Ahwad for giving such a nice introduction. I do not know whether I deserve it, but I sincerely appreciate it. So thank you, sir. So today I'll be talking with you on a topic which is very dear to me, and this is on cytotaxonomy, and which, according to us, is an underexplored discipline in the Western Ghats of India. So this would be the structure of my talk. I'll just talk about some of the basics because it is very important to know the fundamentals before we venture into a field like cytotaxonomy. Then I'll just give an idea that how one has to go about if somebody wants to pursue this branch, particularly for young botanists, budding botanists, how one can go. And then I will tell you some of the examples, classical examples. Mostly, I will try to cite our own examples, which we have worked out in our laboratory in Shivaji University, Kolhapur. And then I'll just give an idea of the scope for this kind of work in a place where we work. So, all of us who are staying in this part of the country, Maharashtra, we all are near to Western Ghats. So, I'll try to tell you that what can be done in this interesting field of cytotaxonomy. So, there is one very famous quote or a dictum which was given by Hitoshi Kihara, and this dictum or this quote, it reads that the history of Earth is recorded in the layers of its crust, and the history of all organisms is inscribed in the chromosomes. So this is a very pertinent quote, and it still holds true. It will always hold true. The point is, if you really want to understand organisms, if you really want to understand species, you see, whenever you are working in the field of plants, you end up working on a species. Whatever you will be working on, you will be working on a fundamental unit of taxonomy, that is a species. If you work on a genus, you end up working on a species. You work on a family, you end up working on a species. So if you really want to know about species, if you really want to know about organisms, how they are related to each other, what they are, what is their identity, there some chromosomal information is something that you cannot shy away from. And before I go ahead, I just want the young generation to know that Hitoshi Kihara was a Japanese professor, and Hitoshi Kihara is to Japan what Nikolai Ivanovich Vavilov is to Russia. So all of us know about Nikolai Ivanovich Vavilov. He gives the centers of origin of crop plants. Hitoshi Kihara was equally important, and his contribution has been very significant. He was the first person to tell that in the hexaploid bred wheat, you have three genomes A, B, and D. So Hitoshi Kihara is an equally important name in the field of genetics and evolution. Now, for the younger lot, I want you to understand that there are certain things, certain basics you should know. So, all of you know about taxonomy. Taxonomy basically deals with identification of plants, giving them correct name, classifying them in a proper classify them properly based on their natural affinities. Then there are two words which one should use very carefully. There are two words. One is called as cytology, another is called as cytogenetics. So cytology is a broad word which talks about anything you are doing for cell. If you are studying a cell, it is called as cytology. So anything related to cell becomes cytology. But when you talk about cytogenetics, you are basically talking about nucleus. You are talking about that organelle of the cell that is controlling the genetics of the cell. So that is nucleus. So you are basically talking about nucleus. You are talking about chromosomes. And cytotaxonomy is a kind of, you can say, combination or a marriage of taxonomy and cytogenetics. It is not taxonomy and cytology. It is taxonomy and cytogenetics. So when you amalgamate, when you mix taxonomy with cytogenetics, then it becomes cytotaxonomy. So you can say a cytotaxonomist look for chromosome characters which can help him or her in delimiting the fundamental unit of classification or taxonomy, that is a species or any category above a species. So cytotaxonomy is utilization of chromosome data. You are using cytogenetical information. And now what has happened? This is something which is not new. This has been already pointed by many people in the past also, that it is very difficult to do field work and be equally good in preparing chromosome squashes or chromosome analysis. So it's a very difficult thing because field work itself takes a lot of time, it requires lots of expertise, and it is very painstaking. And then doing chromosome analysis, preparing chromosomic squashes, both things can be very, very difficult to do. But this is something, this kind of combination, although it is very difficult, but if young botanists strive to master these two disciplines, sky is the limit. A lot can be done in botany, and as I'll show you in the coming slides, that you can do good research. You can provide original ideas. You can prove many things, provided you have the expertise of these two things, and that too without the need of any kind of highly sophisticated instruments. So before I go ahead, as I told you, chromosomes are like, if you want to know about organisms, you should know about chromosomes. I also told you that these two skills are same. But for a person who is working in the field of taxonomy, or for any other person, you must also acknowledge another thing, that chromosomal information is not the only information that is useful for deciding the identity, or the for delimiting or circumscribing a plant group. So don't think that chromosome information, you should not take an impression from my talk that rest all other branches are not important. Only what is important is cytotaxonomy, or only chromosomal information is important in resolving taxonomic dispute. It is not that way. So I also want you to tell that it is important. Many a times it has been useful. It is useful. It can be very cost effective, but do not take an impression that whatever I am telling you is the ultimate thing, ultimate information which can help you in taxonomy. There are various other branches that can help you. But today I will be highlighting the importance of cytotaxonomy, chromosomal information. So chromosome number is very important. It is equally important as a morphological data. Okay, so don't carry a notion that chromosomal characters are everything. If you want to do taxonomy, if you want to understand species, they are the one and they are the one and only important things you have to look into. Now, cytogenetics, now it is done. There are three phases that have come in cytogenetics. The normal one, which you do in your classes, you make routine squashes of onion tip. That is called as cellular cytogenetics, where you use a stain like carmine or orcine and see the chromosome. Then comes molecular cytogenetics, where you are using a DNA probe, and this DNA probe you label with fluorescent dyes, and you try to hybridize that probe on the chromosome preparations, on the squashes. We call this as in situ hybridization. And then final thing, which is very, very popular, is called as flow cytogenetics, where you are going to use flow cytometer, an instrument called as flow cytometer, and you try to estimate the genomic DNA content of an unreplicated haploid nucleus. So this is one example of in situ hybridization. There are two images which you can see. One on the left is fluorescent in situ hybridization, which is done on the chromosomes of hexaploid wheat, which has got 42 chromosomes. And on the right side, you are seeing two nuclei and one chromosome preparation. And you can see two colors. One is a green color, which is given by FITC biotin, and a pink color is given by the combination of digoxygenine and rhodamine. So the second technique is called as genomic in situ hybridization. Fluorescent in situ hybridization, you use only a specific portion of the DNA called as probe. You label it with a particular dye and you hybridize it on the chromosomes. But in genomic in situ hybridization, you take an entire genome of an organism and you hybridize it on the another organism's chromosomes. So now if you see, you will come to know that this is a preparation of spring onion. And spring onion is a hybrid onion which is formed by the cross of Allium fistulosum which has got 16 chromosomes, and Allium tuberosum, which has got 32 chromosomes. So you can see, even when you do not have chromosome in the interface nucleus also, you can see two genomes which are labeled with two different colors. And if you see the chromosome preparation, you can say that spring onion, 16 chromosomes, they have come from tuberosum, and 8 chromosomes which are pink in color, they have come from fistulosum. So this technique of in situ hybridization, and particularly genomic in situ hybridization, is very, very important if you want to ascertain the ancestors of a hybrid. If you want to know the descent of a hybrid, how a particular hybrid has formed, what are its parents. So this technique is very, very important. Okay. Then the next one is something called as flow cytometry, and you would not believe that this flow cytometry is so popular now. If you go on the Q website, there is a data called as C value database. You can see one database on the Q website which is called as plant DNA C value database. And what we do in this technique is you extract the nuclei from the leaves sample or any other sample, and you stain them with a particular dye, and with the help of a laser beam using a flow cytometer, see you try to see what is the how the life is getting scattered, and based on that you get some certain peaks. And once you know that once you have the values of a standard plant, you can find out the value of the plant, C value of a plant you are not aware of. So this is a very powerful technique. With the help of this technique, you can find out the genomic content, DNA content of a haploid nucleus. And it is so popular that there is now a database. And it's not only for flowering plants. You have C value data, C value for gymnosperms, stridophytes, and as of now, more than 10,500 flowering plants have been screened for their C values, and you would be very surprised to know that in case of flowering plants, which are the most dominant group of plants on the earth, which acquire 95% of the earth, 95% of the 95% of the plants on the earth are flowering plants. You would realize that they show a huge variation. There is 21,000 fold variation, with the smallest 1C count recorded in Genlecia aurea, which is a member of Utricularia family, Lentibulariaceae. It has got 0.07 picogram, 1C value of 0.07 picogram, whereas in a Liliaceae member, which has got huge chromosomes, the value is 152.23 picogram. So there is a tremendous diversity, and this tremendous diversity shows how different plants are, they are adapted to different situations, how different habitats, how varied they are in terms of their habitat, in terms of their characters, morphology. So this also is very important. Now, having said that, there are three levels at which cytogenetics work: classical cytogenetics, which we call it cellular cytogenetics; molecular cytogenetics, that include in situ hybridization like fish and gist; and flow cytometry or flow cytogenetics, that involve the use of flow cytometer. The most important or most sought after, or you can say most easily done phase of, or the type of cytogenetics is cellular cytogenetics. Although all over the world techniques have advanced a lot, and people are doing a lot of molecular cytogenetic studies, they are doing fish and geese studies, they are doing flow cytometry studies, but maximum data today also comes from cellular cytogenetics. And whenever we talk about cellular cytogenetics, the most important character is called as chromosome number. What is the chromosome number in the root tip? What is the sporophytic sporophytic number? So this chromosome number is the most widespread and cheapest systematic character. Cheapest because it is very cost effective. You need not to do a lot of things. You require very less instruments, chemicals, very few chemicals are required. High-end instruments are not required. So although there are different phases of cytogenetics which are occurring, But if you see, the most important phase or most important branch is still today is cellular cytogenetics that can contribute to resolving taxonomic disputes. And this is such an important thing that nowadays people have started making chromosome number databases, because uh, it is the era of online information. So you have chromosome number databases on Russian mammals. You have databases particularly focusing on a particular family, particularly focusing on an important genus. So, for example, for cardamine, there is a separate. Cardamine is a Brassicaceae member. Different countries, they are also trying to make their chromosome database for their flowering plants or non-flowering plants. So this chromosome data, you cannot undermine its value. It is very important. On the similar line, when this was going on all over the world, in India also many people took initiatives, and in 2013 there was one grant that was given by DVT to 4 to 5 universities to run a multi-institutional project to build up a certain kind of database for Indian plants also. India has got 2 biodiversity hotspots, a lot of rich, we are a species-rich country. So for India also, it was tried that there should be a database. So one database has been made for Indian plants also. So this also you can see, and this database is not only about chromosomes, it also gives you data on other uh, details also. It also tells you about fingerprinting data, whether it is available or not. Is there any evolutionary evolutionary phenomena, unique phenomena that occurs in a particular plant. So this data is a very thorough data. Unfortunately, it could not continue. There were 2 phases, but now whatever we could do in 2 phases, 6 years, that is now hosted by Botanical Survey of India. In the coming slides, I'll give you the link of that. You can go through. So making database is also very important, because it is a kind of permanent storage of data which can be easily disseminated. Anybody can access it. Now, coming on to what exactly cytotaxonomy involves. I told you cytotaxonomy, it can include cellular cytogenetics, it can include molecular cytogenetics, it can include flow cytogenetics. But as I told you, the most widespread and cheapest and easy to do is cellular cytogenetics. So most of my slides will now be focused on cellular cytogenetics. So what particularly a cytotaxonomist has to do? So taxonomists, they are always after delimiting species, circumscribing species. What is this? Whether this has to be called as a different species or a variety. So they are always into the identity of the organism. So simply, if you want to define cytotaxonomists, they use chromosome features. They observe important chromosome features to resolve the identity of the plant. So first important character that is used, cytogenetical character that is used in taxonomy, is called as chromosome number. This chromosome number can be denoted in 3 ways. You can call it as gametic number. If you call it as a gametic number or a haploid number, you denote it as N. So for knowing this number, you have to do meiosis, particularly male meiosis we do in plant. Second, the most common one is called as zygotic number or sporophytic number or diploid number. So most of the time what you do is with the onion root tip is this. You try to find out the sporophytic number. And X is called as the base number or the basic number. So if you see Triticum aestivum, so Triticum aestivum is a hexaploid wheat which has got 42 chromosomes and its base number is 7. So you can say 7 chromosomes are repeated 6 times. That's why it is called as hexaploid. Is that clear? Then if you see, there is a variation of chromosome number is a very important criteria, and it is very important in taxonomic verification or taxonomic identity. If you take a survey of variation of chromosome number in plants, you will find that chromosome number varies a lot. So in entire, if you see flowering plants, chromosome number ranges from 2n is equal to 4 to 2n is equal to 640. So 2n is equal to 4 is only found in 6 species, and two of them are Asteraceae member. One is a grass, one is a Cyperaceae, another is a Hyacintheraceae or a Liliaceae members. And the highest one is found in a succulent called as sedum. There is another palm which is also a monocot, which is also having a huge number, which is more than 550. But the maximum is 2n is equal to 640. This is the limit in flowering plants. And if you see across the orders Asparagales, which includes all your lilies, crinums, hymenocallis, allium, everything, they show the maximum variation. The minimum is 4, maximum is 228. Orchidaceae, which is a very large family, the variation is from 12 to 240. And in Asteraceae also, the number can range from, 110, that is 220 to 240. So these are some of the plants which are record setters. So I told you there is a plant which has got only 4 chromosomes. This is Ornithogallum. Then this is a palm which has got more than 550. So this is a preparation which was made by a person. This is a an example of a fern. This is not a flowering plant, so I did not discuss this. So it has got n is equal. It has got 630 bivalent. So total 1260 chromosome. This is the highest in plant kingdom. But even in okra, even in your lady's finger, the number is very high. It is 2n is equal to 200. So in plants, see another member. This is a Asteraceae member which has got 2n is equal to 4 chromosomes. And then the lowest in the living world is an ant which is called a jack jumper ant, which has got only 2 chromosomes. So both this 2n is equal to 4, which is found in a Asteraceae member, and this is an ant. Both these they occur in Australia. Why these organisms with lowest chromosome occur in Australia, we have no idea. But my point of telling you why I am focusing on the importance of organisms with a lot of variation in chromosomes, and particularly the organisms with less chromosome numbers or very low chromosome number, because these are plants, these are organisms which can serve as model organisms. You can test many hypotheses. They can serve as a model plant to know how chromosome structure and function is evolving. So these plants with very low chromosome numbers are very important. Then the second important character after chromosome number is the total length of the chromosome complement. So if you see, lotus is also a Fabaceae member, VCI is also a Fabaceae member, but you see the chromosome size. So within a family only, you get huge variation. So Leguminosae, there are 2 genera belonging to Leguminosae. There is a huge variation in the chromosome length and the total DNA. So this is also a very important thing. It gives you a direct clue about the genome size of a particular species. Third important character which a cytotaxonomist look for is that how the chromosomes are staining. So staining also depends on how much the DNA is having a euchromatin and a heterochromatin. Heterochromatin is a repetitive region which is inactive in gene, and that takes a lot of stain. But there are certain families like Malvaceae and other which are very difficult to stain because chromosomes are small and euchromatic region is more. So that thing is also important. So all your Amaryllidaceae, Hyacintheraceae, Liliaceae, they stain well. They take up stain well because they have a lot of repetitive DNA. They have a lot of heterochromatin. But if you come to Malvaceae, it is very difficult to stain them because they have got a lot of euchromatin. Active regions are there. Genes are more. So this also is an important character. Then chromosome morphology. So chromosome number, at times when you are investigating if you are investigating species of a same genus, it may happen that the chromosome number may be same. Now, how do you differentiate when the chromosome number is same? You have to look for chromosome morphology. So based on the position of the centromere, you can decide the chromosome morphology. So chromosome morphology is equally important. When you cannot delineate species of a genus by means of chromosome number, you have to look for chromosome morphology. whether the chromosome morphology is same or different. So this is the fourth character. Fifth character is relative chromosome size. Okay, so within the same family, within the same order, you will find lot of variation. Now see the figure on the right side, you see Nymphophia. They all are Liliaceae. Broadly, you can Sensu, Leto, they are all Liliaceae. Dracena, Draco, you see Aloe, you see Yucca, and you see Gasteria. If you realize, you will find that in Aloe, you see what is called as a bimodal karyotype, and or I'll talk about bimodal karyotype later on. You can see in Aloe, you can divide the chromosomes into large subsets and small subsets. But in Nymphophia, this is not the case. In Dracena Draco, this is not the case. Gasteria, again, you can say four large subsets and three small subsets, three small chromosomes. So this also matters. Chromosome complements of most species of plant, they consist of chromosomes of comparable size. But when you go across genera, when you go across family, these are different. Okay, sometimes within the genus also, I will show you some of the examples where within the genus, species of a same genus, they may also show different kind of chromosomes, chromosome sizes, and in fact different karyotypes. So this also is something that has to be observed, and it is very important for deciding the limit of a species. Another very important thing is you have to study meiosis. Why meiosis? Meiosis is important because it tells you about the behavior of the how the chromosomes are behaving, whether they are pairing, whether they are segregating properly. So you can have abnormal meiosis, you can have a proper meiosis, you can have chromosomes pairing, you can have chromosomes which are pairing and immediately falling apart. You call it as desynapsis. You can have chromosomes which are not at all pairing. That means they are not homologous, they are showing asynapsis. So all these things that help you to understand the relationship between species. In taxonomy, there is a branch called as biosystematics or experimental taxonomy, where one of the most important uh, aspect to find species relationship is to hybridize species and to observe the meiosis in the F1 hybrid. So if the F1 hybrid between two different species is showing a proper meiosis, that means those two species are very genetically close. If it is showing highly erratic meiosis, abnormal meiosis, you say that these two species are genetically very distinct. So all these characters 1 to 6, whatever I told you, that has to be studied by a cytotaxonomist. And all these 1 to 6 characters, you can broadly call them as karyotype. They all are called as karyotype. So if you want to put it in nutshell, you say that cytotaxonomists are more focused on karyotype. They use karyotypic information to delimit species. So this concept of karyotype, this was given by Levitsky. I will not read through the definition. All of you know about this. So karyotypes are usually, this is the standard way how you depict karyotype. And what is very important is that karyotypes are mostly species specific. That is why taxonomically they are diagnostic, they are very important. Each species has got a specific karyotype. So whenever we study karyotype, the standard way of organizing a karyotype is in a descending manner. So the largest pair will come at the first and smallest at the last. Short arm should point upwards, long arm should point downwards. So this is the way it has to be depicted. Once you make once you have done it, you can show it as a karyogram and ideogram. Usually, whenever we prepare a karyotype, most of the time we use a tissue called as root tip. That is a easy tissue. It doesn't have chlorophyll and all other disturbances it doesn't have. But sometimes in case of those plants where root tip is, for example, there are root parasites, there are plants which have no roots, there you can study leaf tips, stem tips, tendril tips, or meiosis for doing the karyotype. But most of the time, karyotype information is produced or is obtained through a root tip metaphase. Okay, karyotypes, if you see, as I told you, there are 3 lakh to 4 lakh species. I mean, whatever data we have, flowering plants are about 3 to 4 lakhs. We do not know much about the chromosomes of all these species, but whatever we know, that based on that, karyotypes can be called as symmetric karyotypes. Karyotypes can be grouped as asymmetrical karyotypes. Asymmetric or symmetric karyotypes, they differ in the type of chromosomes they have. So a symmetric karyotype is a karyotype which has dominance of a particular, of one type of chromosome. So they would usually have submetacentric or metacentric chromosomes. Asymmetrical karyotype will have different kind of chromosomes. So it will have a metacentric also, it will have a telocentric also. Same is the case with this also. And it is believed that in evolution, asymmetric karyotypes, they are advanced. But let me tell you that evolution does not happen in a defined manner, or you cannot define it. Sometimes a symmetric karyotype could have arisen as a result of an asymmetric karyotype. So in which direction the karyotype is evolving, it is very difficult to say. You cannot always say that symmetric karyotype is primitive or it is a primary condition, whereas asymmetric karyotype is a derived condition. Sometime asymmetric condition leads to the derivation of a symmetric karyotype. So both way it can be true. You have to analyze a particular group, and then you will have to observe many things. You have to understand the data, interpret, and then only you will come to know how the karyotype evolution has been. In addition to this symmetric and asymmetric karyotype, there are a very specific category of karyotype which is called as bimodal karyotype. You can call bimodal karyotype as a special kind of asymmetric karyotype in which the organism has, the species has two types of chromosomes, large ones and small ones. So you can see this is one very good example. Dipkadi is a very common material which you will find in Mumbai and this part of the country. It's a bulbous plant. You can see there are two pairs which are very large. Remaining all other pairs are very small. So when there is a discontinuous type of karyotype, or you can say the difference in the length of the large chromosome and small chromosome is too much, you call it as bimodal karyotype. So these are very specific karyotype. Nowadays, a lot of work is going on these kind of plants. People want to know, why is there any evolutionary implication of having two types of chromosomes, large ones and small ones. So this is now a thrust area. And in our part of the country, Dipkadi is such a plant which is very important, where you find this kind of karyotype. Now, apart from normal chromosomes, which you see A chromosome, sometimes you can also observe B chromosomes. I will not go into the B chromosomes. B chromosomes are also a thrust area nowadays, because initially they were considered as inert. B chromosomes are different from A chromosomes in the sense that they do not follow Mendelian inheritance, and their loss does not create any kind of problem to the plant. But earlier they were thought to be providing some kind of ecological adaptation to the plants in which they are occurring, and they were thought to be inert. But now the concept is that B chromosomes also carry gene, and B chromosomes can be modified, they can be engineered, and by engineering B chromosomes, you can introduce some desired character in crop plants. So B chromosome also is a very important you can say subject which is being taken up by people in this field. So B chromosomes are also very important. So we have also observed B chromosomes in some of our plants which we work on. I will not go into the detail. So having said that, what a cytotaxonomist does, it is also very important how to do it. What is the methodology? So there is no rocket science in methodology. Your teachers teach you everything. Everything is there in the book. What is very important is to select a group of plant. Now, when I say select the important group of plant, is the plant should have some kind of taxonomic dispute. There should be some taxonomic ambiguity. So that kind of plant you should select, so that you can use chromosomal data to resolve its identity. And you need to understand how this plant grows, how you can harvest a good tissue which will have a lot of division. So all these things are very important. The most important thing is selection of plant. So selection of plant, see, I have been showing you, you must be seeing lots of metaphase spreads, which are my own, unless they have been, indicated. And it might appear to you that it is an easy thing to do, but in many plants, even we are not able to do, because they are very difficult to work on. The root tip may not be hydrolyzing properly. The chromosome size would be very small. They will have a lot of euchromatin in them. They will not stain. So it is not easy. So the step of selecting the plant group for doing cytotaxonomy is very important. It is the key step. Okay, once you have done it, you understood it, you should know how that group can be maintained, how it roots. So if you want to plan a kind of cytotaxonomy study for your research, it is better to work on easy plants first. If you start altogether with a plant which is very new, for which there is no standard protocol, it may happen that you will never touch the microscopes, and you will never take the stains again. And that is what happens, that is what is happening all over the country. Now you will not find people who are sitting and looking into the microscopes and observing chromosomes. Why they are not seeing is because the plant material which they have taken, somehow it didn't show the result. They could not see it, so they didn't find it very interesting. So once you started seeing them, you get an interest. So it is always better to start or learn this technique of making squashes on groups which are easy. And some of the very easy groups which are easily available in the nurseries or in the wild field in this part of the country are these plants. These are plants where chromosome numbers are also very less, and their size is also very good. For example, chlorophytum, the plant which is on the extreme right. So this is a cultivated species on the top, but the species which is at the bottom right is a species called as chlorophytum lexum. This is a species that occurs throughout our part of the country, Maharashtra. You go to any rocky place, you will find it, and it will come during this monsoon. So it is very easy to maintain. It has only got 16 chromosomes, just like your onion, and maybe slightly smaller than onion. So onion has got a chromosomes which usually entangle, because they are very long. So this has, in my opinion, I would say this is a very good material, at times better than your onion, because it is very it shows lots of divisions. You get good root tips, and the chromosome size is absolutely the way you want it to be, very optimum. So if you work on these groups, maybe you will develop interest. Then there have been various examples of taxonomic utility of chromosome data. One of the classical examples all of you must have read is an example, or is a study which was done by McKelvey and Sachs in 1933. Though the plant on the top left is yucca, and at the bottom left is agave. So yucca was put in Liliaceae, and agave was put in Amaryllidaceae. Yucca was having an inferior ovary, and agave was having a superior ovary. So coronary series is where you have Liliaceae, which is a superior ovary. Epigyne is a series where you have Amaryllidaceae, which is having inferior ovary. So they were put together in separate families. But then studies were done, and this is a dikinesis. You can find that there are 30 bivalent. This is a dikinesis. This one is a metaphase first, and this one is an anaphase first. So if you see the dikinesis here, on the left it is yucca, on the right it is agave. You will find that both yucca and agave, they have 5 pairs of large chromosomes and 25 pairs of small chromosomes. So McLeave in 19 and Sachs in 1933, based on this kind of karyotype, they proved that yucca and agave, although they have different ovary arrangement, there one ovary is inferior, another it is superior, they should belong to the same family. So today they belong to the same family. Even the latest classification, APJ4, treats them in the same family, which is your Agaveaceae. They are now in a part of the same family, Agaveaceae. So they have been put together. So this was done as early in 1933, just based on the kind of chromosomes they have. So such a important information these chromosomes tell you, right? Another very example is about Senecio. Many species of Senecio have come as many species of Senecio, they have arisen as a result of hybridization. Senecio is a classical group. where hybrid speciation has been studied. And this hybrid speciation has been proved by chromosome studies. So, for example, if you see on the left, this is Squalidus, Senecio squalidus, and Senecio vulgaris. Senecio squalidus, it has got 20 chromosomes. Senecio vulgaris, it has got 40 chromosomes. They hybridize in nature. They form a sterile hybrid, which is 30 chromosomes, which gets diplodized. And finally, you get a hybrid species, which is having 2n is equal to 60. This is a classical example of hybridization. So this has been possible only because of chromosome study. So in nature, people find this plant and then they did the same thing in the lab. So chromosomal information, chromosome preparation can tell you if species are formed as a result of hybridization. So it's a very unique, tool to tell you how species are getting formed. Now I'll give you some of the examples which we have done in our lab. Some little examples. We haven't done much. A lot has to be done. So there is a group of there is a family called as Colchicaceae, which is known for is for an important alkaloid colchicine. All of you know about this. And this Colchicaceae, in our part of the country, there are only two genera in Colchicaceae. One is Iphigenia, another is called as Gloriosa superba. So in Colchicaceae, there is a tribe called as Iphigenie, and this tribe Iphigenie is distributed in India and Africa. And there are only two genera, Iphigenia and Camptorrhiza. And how they differ? So this is the distribution of Camptorrhiza. So Camptorrhiza, you can see it is there in South Africa, southern part of the African continent, and it is shown in India also. Iphigenia is distributed in India, Australia, New Zealand, also in Africa and Madagascar. Now, in somewhere in 19, so there are two genera, Camptorrhiza and Iphigenia. Camptorrhiza is only there in Africa and India. Iphigenia is there in Africa, Madagascar, New Zealand, Australia. So the genus Camptorrhiza, if you see the etymology, if you try to understand the etymology, you will find that Campto means curved, rhiza means root. So roots are curved, knee-shaped roots. And if you see the flowers, if you see the filaments at the middle, they have a nectary. So this is Camptorrhiza strumosa, which is found in Africa. Okay, and this is Camptorrhiza indica, which was described by my boss somewhere in 1992 or 93 from Ratnagiri. It was that time called as Camptorrhiza indica. Now, if you see in Camptorrhiza indica, the style is undivided. So when this species was described, style is undivided. It was called as Camptorrhiza indica. These are remaining species of Camptorrhiza. So see, this is Iphigenia. This is Iphigenia indica, magnifica, mysurensis, pallida, and stellata. All have trifid style. Camptorrhiza indica, when it was described from India, why it was not called as Camptorrhiza? Because it was not having a tuber-shaped, knee-shaped tuber. It was having a globose core, and also it was having undivided style. Okay, so Indian species was described as Camptorrhiza indica. But when we conducted our studies, we found that Camptorrhiza indica that was described by Professor Yadav as Camptorrhiza, it is absolutely similar in its cytogenetics to the genus Iphigenia. So we did chromosome analysis, everything came out to be very similar with Iphigenia. Then we did phylogenetic analysis. We found that Camptorrhiza indica, which was described by Yadav, is sitting just beside, Iphigenia stellata. Then we found that Camptorrhiza indica is only African. Indian Camptorrhiza should not be called as Camptorrhiza. It should be called as Iphigenia ratnagirica. We did not stop there. We wanted to see whether they really also are same, Iphigenia and Camptorrhiza indica described by Professor Yadav. So we crossed Iphigenia stellata with Camptorrhiza indica or Iphigenia ratnagirica, and we found that they cross, and the hybrid shows perfectly normal meiosis. So all these studies tells you that chromosome data through hybridization, through meiosis, through metaphase spreads, karyotype, it tells you that Camptorrhiza indica that was described from India is not at all Camptorrhiza. It should belong to the genus Iphigenia, making Camptorrhiza a monotypic genus confined to Africa. So this study was very well received, and it was published in a good journal, Journal of Systematics and Evolution. Then another very important group which we worked on is Flamingia. Flamingia is a wild relative of pigeon pea. Okay. And I will not go into the detail. Wild relatives are very important because they are the source for broadening the genetic basis of crop plants. They confer disease resistant traits and other traits to cultivated plants. So this is one group which we worked on. And we found that in this group that none of the species has a variable chromosome count. All have 22 chromosomes. So cytogenetically, they are very uniform. So now you would be realizing that when you go from a monocot to a dicot, legume, from liliaceae to legume, the size of the chromosome, staining of the chromosome, preparation of the chromosome, it changes. So that makes this branch very challenging. So wild groups are very important. Wild crop relatives are very important. Another important genus is called as Rankanda, which is Dremia, very common in our part of the country. Taxonomy is very difficult. All bulbous plants are very difficult to identify because they would flower only for one or 2 months during monsoon. Rest of the season they would be photosynthesizing. They will have green leaves, and after October till next April, they will be hibernating. They will undergo dormancy. All above ground part will be lost. So that is their strategy. So identifying them is very difficult. Herbaria will have their poor specimens. Flowering happens only for 10-15 days, very rarely for a month. And during that period, if you do not visit them, you will encounter them in their vegetative stage. So one such genus is called as Dremia, which is very difficult to identify. So different kind of flowers are there. I will not go into the detail. Time is also running. So we tried to work on this. Morphologically, these flowers, they look very similar and it is very difficult to delimit them. But when you do chromosomes, you find that they differ in lots of details, the chromosomal details. They can differ in chromosome number. For example, there is one very widespread species, chromosome Dremia indica, which although it shows 30 chromosomes, some forms show 30 chromosomes, some show uh, 20 chromosomes, but they differ in their karyotype. So one of the forms which we collected from coastal area is a triploid. It shows lots of anomalous behavior. So it tells you that Dremia indica is a species that shows lots of cytogenetical variation. So how species vary? So this also helps you in delimiting species in a group which is highly variable. Okay, so if you do this kind of study, you understand the extent of variation a species can show. So we did meiosis also. This is a very good material, and we found that different species can be differentiated using these characters. So we did crossability studies also. This is emasculation. You can see anthers have been removed. They have been pollinated with the pollen of other species and they have been bagged. So finally, we build up a crossability polygon. This tells you the relationship between. So green crosses are successful. If two species they cross successfully and their hybrid is fertile, you say that they are genetically very, very similar. So these kind of things you can understand about the relationship of the species if you do cytotaxonomy. Then this is one example where we made an artificial hybrid. We crossed Dremia govindapi with Dremia coromandeliana. One is with 2n is equal to 20, another is 2n is equal to 40, and the hybrid was a pseudo-diploid with 2n is equal to 30 chromosomes. So these kind of things also you can do. So this is the magic of experimental taxonomy. So then you prepare comparative karyotype. We have studied this for Barleria. Barleria is very ornamentally important genus. It's a wild ornamental, and if you have to catch its potential, if you want to uh, utilize its potential in horticulture industry, you need to know its cytogenetics. So we have worked out this cytogenetics. We know which plant is a good female, which can serve as a good female parent, which can serve as a good male donor. Aponeridon is another group which is very tricky, and there are many species which do not set seeds through. Which do not set seeds, which have reproductive constraints. So we wanted to know. This is one example. This is a species described by Professor Yadav from Kudal region, coastal area. He described the species, but he noticed that the seed set is very low. So we wanted to know why the seed set is low. We did meiosis. We found a lot of anomalies, and we could say that this is a species that has been formed as a result of hybridization. Although we do not know what two species hybridized, but this is a hybrid species and it is not stable, and meiosis is highly erratic. Similarly, there is another widespread species, Aponeton natans. It shows almost 100% seed set. Every flower, every carpel in the flower, it converts into seeds. So we saw meiosis was completely disturbed. And when meiosis is completely disturbed, it is showing huge amount of seed set. So then I think it is also reported in the literature. So this is an apomect. Same is the case with Aponeton undulatus. It does not set seeds. It only produces bulbils. Okay, it vegetatively propagates. So we want to investigate why it is happening, why it is not setting seeds. So then also we realize that the meiosis is highly erratic. There is laggard formation, there are bridges, there are precocious separation. So meiosis gives you a chromosome behavior here is giving you an idea about the genomic stability of the organism, of the species. So another thing is we have studied the taxonomy of one species which was not clear through morphology. I will not go into the detail. This is one group which we are currently working on. This is a very important group. It could serve as a model plant. We are working on this. This is a plant which shows three different kinds of karyotypes: bimodal, trimodal, and unimodal. So you can see very good material. Chromosome number is also very less. So you can do very good kind of studies, particularly molecular cytogenetic studies using in situ hybridization can be done, and you can understand how chromosome structure and function evolves with reference to ecological conditions. So how do you start? How do you start from scratch? If you want to do cytotaxonomy, there are certain websites. Now, this is a very important website. If the young people are there, they may take a note of it. This is a site which is maintained by Tel Aviv University in Israel, even though they are most of the time in war, but they try to see that this website is working. It gives you, first-hand data about how many plants have been worked out. You get an idea about the plant. Then there are some Indian books which you can see, but this is a very good book, a good website where you can check whether this plant has been worked out, what are the relevant literatures. Then Missouri Botanical Gardens, St. Louis, they also host a website. It is there along with Tropicos, and they call it as Index to Plant Chromosome Number. Here also, if you write the name of the species, it gives you what is the sporophytic count and the corresponding reference. So this also is good for searching the literature. And this is what I was telling you, that the DBT has given us DVT gave us some funds for 2 years, two phases. So whatever data we could generate for pteridophyte, gymnosperm, and angiosperm, it is now available. You can visit this website. It is now available on the BSI link, and you will get an idea. You will come to know what are the plant groups you can put your hands into, and it would be very useful. And why I am advocating cytotaxonomy is that still less than 30% of the flowering plants are untouched. Many new things you may discover. Western Ghats is a very good place to work because almost more than 2,200 species are strictly endemic. Okay, we do not have any information on their chromosome number. If you see what groups you should work on, groups which are wild crop relatives, wild ornamentals, economically important groups, particularly those families which are huge in size. For example, Poaceae, Asteraceae, Eriocolaceae. These are families which have gone tremendous evolution. They show evolutionary peak. So finding good novel findings in these groups, uh, chances of novel findings are more in this group. And another important thing is endemic taxa. Endemic taxa are confined to a particular location, so it is very difficult for any other person to work on it. A person sitting in Himalaya cannot work on the endemics of Western Ghats. So it has to be done by somebody who is sitting in the Western Ghats. Okay. And this is my last slide. Uh, this is for youngsters. I'm not teaching any teacher. So this is for the youngster that do not get carried away by the, high-end instruments, highly sophisticated instruments. They are required, but conventional studies are very, very important. And your studies and the kind of instrument you are going to use, technique you are going to use, it should be based on what plant group you are working on. You should not decide the instrument first and then choose the plant. Once you take a plant group, then you can decide what kind of work you want to do, what kind of sophisticated instrument techniques you want to use. So your investigation should be taxon based, they should not be technique based. Thank you. I thank all of you for the patient hearing. I thank all my uh, students who have helped me in whatever little we have done in our lab. Thank you.
Thank you very much,
Thank
sir.
you.
And actually, the session is open for a few
Yeah. If
inquiries
I'm,
if they are there. But
yeah,
just
Yes.
I
Anuradha
will,
madam, can
yes.
I speak for a minute?
Yes, ma'am.
Yes,
Yes, yes.
just
Yeah,
okay, okay,
yeah.
okay.
Dr. Manoj
Yes.
Lekhak,
Yes,
you,
ma'am.
at least for me, you kept on saying young, young, but you took me back into a very nostalgic journey. I remembered plant chromosomes, Archana Sharma, then Darlington, and amazing, but it's painstaking, it is not easy.
Yes.
Onion root tip, garlic root tip is okay. But even to get a good spread, proper fixation, and especially meiosis, amazing, amazing slides. Hearty congratulations. You made us very proud. And Dr. Yadav, I'm sure, must be very, very proud of you. You being a student and excellent. And I'm sure you're a very good teacher.
Thank you,
Like,
ma'am.
Yes,
Thank you.
even for a
absolutely.
minute, I didn't want to miss your talk. And अमेजिंग स्टेनिंग परफेक्ट लाइक ऐसे बोलते हैं ना बच्चों को कि ऐसा होना चाहिए प्रिपरेशन स्पेशली meiosis. But I have one question for you.
Yes, please, ma'am.
You must also, where if possible, you write down. It is not very easy to even root tip, especially grasses and all that. They are not easy like onion जो ऐसे smoothly hydrolyze हो जाते ऐसा नहीं है.
Absolutely.
And the type of pre-treatment you require, maybe we will also request you for one more session, especially for those who are interested in cytotaxonomy and karyomorphology. And another very important thing is if they are ephemeral, see you said dipcardia, their flowering is only in a particular season. And in that season, to get proper pollen mother cells, that means you need that type of expertise. नहीं तो you'll
Yes.
end up either saying pollen grains or anther wall को लेके बैठ जाएंगे.
Yes.
तो बहुत means I feel I will steal away your slides. So hearty congratulations and tons of blessings and excellent, excellent. There is no word.
Thank you, ma'am. Thank you. So
Yeah,
kind
and
of.
also you please tell like if anybody is interested, small workshop or this
I would be
thing.
happy to do.
Yeah,
Same. Yeah, I'm the
exactly.
first student, huh? Even though you are according to you not young, but I'm still young.
Actually, you should. Actually, ma'am, people like you should. You should tell people to arrange because what happens,
We are
seeing is
ready.
believing. Seeing is believing.
Yes,
If you see, then students start taking interest.
yes,
What has
yes.
happened that when we
Huh.
teach them, we do not come up with the slides, so they do not get interested.
Yeah,
Once you start seeing those things, now you get interested, and this is what has happened. Professor Yadav would sit with me, and he would tell
yes,
you, you are a teacher. तुझे ये आना
correct,
चाहिए कल के दिन तू कैसे पढ़ाएगा सिर्फ
correct.
मॉर्फोलॉजी देख के कैसे पढ़ाएगा तुझे ये भी पढ़ाना है तो करो ऐसे
बराबर है.
हाँ.
So one has
Right.
to be a lifelong learner.
Yes,
Excellent,
absolutely,
excellent.
absolutely,
And
absolutely.
we request anybody who wants to join. We are the first one to have the workshop whenever you are free. Whenever
Yes,
you are free.
absolutely.
This is
We'll
one
do it,
technique
Yes.
ma'am.
people
We,
should know.
we'll
There
do
is
it,
no
ma'am.
two opinion on that. And also you give excellent classes are all, most of the food that we eat is all hybrid from wild
Right,
relatives. So
right,
that
Mm.
point also is very well taken
right.
to that you must take care of the wild. And another plant, Barleria, I am going to talk to you some other time.
Yes, anytime you're
Excellent.
welcome.
Yeah, God bless you. And
Thank
thank
you.
you so much for giving me this much time. Thanks
Ma'am,
a lot.
pleasure is all mine. Pleasure is all
Yeah,
mine.
thank you.
Uh,
Thank you, ma'am.
thank
Thank you.
you.
So there are
Dr.
few more
Lekhak, there are
questions.
some questions in the chat box. So,
Let me
Dr.
just see. Just
Yes,
just a
yes.
minute.
Please,
Let's.
please, please.
Chat box, yes. Thank you, ma'am. Where are the questions? Can you read it? Acha, just a minute,
Yeah,
just a minute.
Yes.
My
There-
question is whether plants
yeah.
have polyteny chromosomes as those found in
Yes.
insects. If so,
Hmm.
see, I have not read about polyteny chromosomes in plants, so really I do not know. To be very frank, I do not know the answer for this. Let me get into. I see this and tell you, but I do not know. I'm sorry. Given the rapid
Yeah. So there is
diversification
one
of
more
herbaceous
question.
flora on the northern western United, how can chromosomal evolution provide help us decide
Neo-endemics
Now,
from
see,
the
this
paleo-endemics.
is a very good question, neo-endemics and paleo-endemics. So let me tell you very candidly that there is no criteria to say anything as a paleo-endemic, foolproof criteria as neo-endemic or paleo-endemic. But usually the criteria that has been used by Constantine and Farwegger, if I'm right, they say that neo-endemics are those species which have high chromosome numbers, and paleo-endemics are those species which have retained low chromosome numbers. So, for example, in our case, if you compare one genus called as Aponogeton, there are two species, disjunct species, Aponogeton satarensis and Aponogeton Dickeri. Dickeri is a Madagascan species. It has got 52 chromosome. It's a neo-endemic, and our Satarensis has got 2n is equal to 26 chromosomes, and it is uh, paleo-endemic. Another thing is neo-endemics are restricted to a particular area. They are not widespread. Okay, because they are yet to spread. Similarly, paleo-endemics, once upon a time they were widespread. Now they have become restricted. So there is no hard and fast. I mean, you cannot say a foolproof thing by which you can say that they are neo-endemic and paleo-endemic. But the concept is paleo-endemic would have low ploidy, and neo-endemic would have high ploidy.
That's good.
Another question is,
No, I think everybody is appreciating the
Yes, sir.
presentation and the wisdom you share with them. Uh,
Yes.
so
So
thank you
thank
very
you very
much,
much, sir.
everybody. And just it's a pleasant duty to me to express the vote of thanks. Okay, so I express my heartfelt gratitude to like sir. It's very inspiring and enriching. And what you told about select the plant and not the instrument is very important for youngsters. Actually, this is the message they can take away from, at least from this lecture. As Usha madam told, your slides were wonderful. They were wonderful. Just wonderful. Just like Yadav sir's slides of the biodiversity. So your lecture definitely tells us how biodiversity can analyze and how we can go further in the, for the APJ system, what we are talking nowadays about. Uh, our thanks from all our collaborating institutes, Maharashtra Vriksha Samvardhini, Department of Botany, RJ College, Dr. Baba Saheb Khardekar College, and Natural History Education and Research for continued efforts in promoting botanical education and biodiversity awareness. Thanks to all participants for joining. Huge crowd was joined today, and I'm really appreciated everybody's joining, because such a large group, the number I could get there nearly about the 75. Now somebody they have left, but thanks to participants actually, and youngsters especially, I want to thank, because they can take away the messages from this and they can select the subject of their choice. So we look forward for your presence in our upcoming programs also. Just I will ask Dr. Awad sir to announce the mandates,
Yes, yes,
yes.
definitely.
So
Yes, madam. Yeah.
over to
Uh,
sir.
yes, to all the nature enthusiasts who have joined today, uh, every third Saturday of the month as well as every third Monday. We usually have one session. For the last several years, we are running this program, and this month's the next Monday, we are going to have one more session. And usually in that session, we bring to you a writer who has written a book related to biological sciences. I'm very happy to announce that Monday, that is 20th of July, we'll have a session by Dr. Abhijit Bayani, who is going to speak on his handbook for the spider, a web of life. On the spider, he has written a handbook, and he is going to share with us uh, the the concept of the identification and the diversity of the spiders. Just now, I have shared one link in the chat box that is about the WhatsApp group. That WhatsApp group, we usually post whatever sessions, monthly sessions we have. Apart from that, we have a channel called as Mission Devrai, and whatever lectures we had so far in the last few years and in the future also, all these lectures, recorded lectures, are available for you to see them. So I'm sure you will spread the message as well, and you'll be joining the group as well. Thank you very much. Thank you very much, Dr. Lekhak sir,
Thank you,
for
sir. Thank
the
you.
wonderful session. It was really pleasure having you, and we are looking forward to this collaboration in the future in
Thank
the
you, sir.
form of
So kind of you. So kind
some
of
of
you.
you, some of the participants have asked about the workshop, and
Workshop.
RG College. Yeah, RG College is definitely a place you can anytime, whenever you are free. Definitely, we can have this collaboration further, and I'm sure people would join there, especially the teachers from the biological sciences. So, thank you once again, sir.
Thank you, sir. Thank you. I'll take your leave. Thank you.
Yes, sir. Thank you. Thank you.
Thank you.